Welcome, everybody. We've got David Augustyniak with us today. And because I know how to pronounce his name, I figured I better do it because I'm sure Leighton and Av would go, "How do you pronounce that?" Anyway, guys, welcome to the Soil Matters show. I'm going to turn it over to Av and Leighton and David, and you guys have a wonderful show.
Thanks, Ken.
Thanks, Ken.
So, Dave, why don't we start by welcoming you, Av? Good to see you again, my friend. And why don't you give us a brief synopsis of what you're doing and where you're going with it?
Cool. Yeah. Thank you both for having me. It's a super honor, and I'm excited to share what I got going on. So over here at the Art Farm, we're a permacultural principle teaching farm that focuses on soil biology, mycology, and community in general. So we want to teach all the arts. Arts is a huge thing too that influences a lot of what I've done through my path. And now we're just opening a community lab to provide our database collection, all kinds of different soil science. We have a bento lab in there so we can start doing genomes. We're about to tap into the MinION and start doing all that RNA stuff. We want to be able to do plant genomes, mushroom genomes, and I'm really into the cactus myself too. So we'll be tapping into like cactus genomes and helping like map some of that stuff out for a lot of the communities where I think there's a lot of misinformation. Me and my good friend are co-organizing the Southwest Funga Fest, so we'll be doing a really cool regenerative community, ecology focused education festival that's coming up in July. And so but on the day-to-day we just cultivate gourmet mushrooms and try to get that good food to the people, right? And that the good information, try to provide what we can, right?
That's amazing. Yeah. Maybe David, do you mind let's almost go with a bit of a basic how are you incorporating some of the permaculture principles into fungi on the farm?
I mean, so as far as like hyper local, I'm always trying to create and stack all my systems. So we're taking all the waste stream from the farm, we're feeding it to the chickens, they're going through it, they're digesting it, adding a bunch of nitrogen, then we're going to feed all that to the worms and hopefully create a closed loop circle with the soil production there. I'm working with all the local farmers, always trying to tap into hyper local, any input that goes into the farm, all the way down to our liquid cultures. We work with local beekeepers to try to get honey that's locally sourced to put into it. Always stacking functions, right? Every input has an output that I think we can utilize it again for. So even down to the mushroom blocks for the goats, once that brown and white rock comes in, it helps break down the lignin in the wood actually, and then it can be used as a fodder for goats. A lot of people don't know that, but I think as far as like growing principles, you're always trying to do the best. A healthy mushroom's a happy mushroom, right? So like your input again is always going to be your output, and so there's always things to learn. But we're doing a lot of preservation as well.
I think I definitely want to follow up on the preservation of. Are you talking about preservation of genetics or preservation of mushrooms in extracts and tinctures and things like that?
Yeah, I mean, we're just starting to get into that now with the lab. I think I've had a different approach. I think I like to gather lots and lots of information for a long time and then take the full approach. I'm into like whole medicine production and things like that. So spagyrics and just being able to recycle. We just got a Extract Craft Pro that's going to be able to allow us to tap into some really cool ethanol extractions, and we're going to be able to recycle and use about 90% of that ethanol. And so, just waiting for the right time for me is what it's been about. I really want to get into the spagyrics, whatever, taking all three essences of the plant. I always felt like when I was making extractions and throwing away all this plant matter, I felt like, part of the medicine's gone. Nothing against the cannabis industry. I'm a full flower kind of guy myself, but extractions definitely play their part. And but again, I think it's all the systems together coming together and creating like this bigger picture that a lot of us are missing. I have a lot of friends in academic, and bless them, and I love them all, but I feel like sometimes in academic we forget that you're a part of that system, and the layers of that system is really what makes it work. It's kind of like I feel about our our healthcare system. Nothing against Western medicine, but I think homeopathy and Chinese medicine and even Western medicine all together are going to save the most lives. I don't think one individually is going to save the most lives. I think that working together, we could create a system that could not be detrimental and not be exhausting our resources and taking away from other places so this place does better. I think there's a lot of layers to it, and having an architecture background has helped me sometimes take a bigger picture kind of view. So I feel a little lucky in that sense.
Yeah, it's sorry, just one last comment. It's David, you've used the language around many layers, and to many of us, the mushroom, what we see is what we focus on. But I think when you think of I'm sure of what you teach, it's all about what's below ground. What we see is probably the smallest part of the fungal kingdom, right? So
yeah, I think Leighton, did you want to say something before I?
Oh, I got so much. But go ahead. You fill in with Av, and then when I can, I'll throw something at you.
Yeah, sure. So, I don't know. I always try to tell people good examples like this, right? We see the tree, and the tree has its system above ground. It has its roots. That whole tree and that roots, that's one giant organism, all right? And so on that organism, you have a piece of fruit, you have a pine cone, you have a flower, right? So that I like to call that the reproductive organ of this large organism, right? It's a huge organism, all of it. And then it has that one little part, but each tree produces either a pine cone, a fruit, or a flower. So that's the reproductive organ. And then inside that reproductive organ is usually a seed, right? So I call that the reproductive cell for life. So there's the organism, the reproductive organ, and then the cell for life, which is usually a seed. You can find it in fruit, pine cones, flowers, all the likes, right? So mushrooms, a lot of people don't understand that the mushroom is just that reproductive organ, right? So a lot of the times that mushroom is fruiting because usually it has the right environmental influence, but like most organisms on the planet, once you're exhausted, maybe you've eaten all the food, maybe you're a saprophytic fungi that's like digesting a piece of wood, right? You just see a piece of wood and a mushroom growing out of it. Well, actually, there's a vast complex of mycelium. If you've never heard that word and you're tuning in, mycelium is like a white hyphal thread that looks like spider webs. Sometimes in the woods, you can lift a piece of wood, you see all this little white stuff down there. That's what we call in the mycological world as mycelium, right? A lot of people, they coined the term recently, the wood wide web, right? And it's the vast network of mycelium that's underground, right? But it's digesting lots of things. It's digesting poop, it's digesting wood, it's digesting agricultural waste. Some people have isolated it to start digesting plastic. Like I've seen somebody train it to digest a cigarette butt, right? So that's that organism. That mycelium is this gigantic organism and network that we we barely, you know, are starting to understand. I think Paul Stamets says every step you take in the wilderness or in the forest, there's up to a mile of mycelial networks under each step that you take, right? So vast organism that we don't see. And then that mushroom, that's a little fruiting body. That's the reproductive organ. And then on that mushroom, sometimes you'll see a gill. If you go to the white button mushroom store, the Agaricus bisporus, it has a gill on it. And then on that gill, it has a little structure called the basidia. And then the basidia has about 3 little basidiospores. And each one of those spores is essentially like a seed, right? It's a reproductive cell. And so when that mushroom, like most organisms, it's going to die, it's going to form this organism. That mycelium is going to weave really tight. There's all kinds of action going on. It's one of the most misunderstood things in nature, I think, right now, is like, how is that mycelium creating new tissue and making that leap? And like, I think the philosophy Peter McCoy told me about a long time is called the Spitzenkorper, right? So they're trying to like understand the Spitzenkorper and what's happening at that mechanism when that mycelium is making that leap, right? And so yeah, we have this vast underground system. It's in the trees. Sometimes it attacks living trees, right? And that's what we call parasitic, which I think is a really misunderstood nomenclature too when we're talking about plants, especially and ecosystems. When we in our intervention have the ability to say that this is a parasitic thing, when it's like, well, maybe this intervention was planned over the last 2,000 years and we got a glimpse of it and we think that it's parasitic. Well, really, it's playing a large 500-year role in this ecosystem. And so it's like funny, you know, people are like, oh, well, this Chinese elm is parasitic. But most of the times I see Chinese elm, it's like right where we put a road in the waterway, you know what I mean? And so, yeah, nature's going to fight back and grow this really invasive thing when you're like running over its networks and like dig up and rip its network and then put this roadway right in the middle of it. I think nature deserves a chance to fight back with a quote unquote invasive or parasitic species, right? Dare we call it those? And so, yeah, I mean, the mycelium is all over, right? I think I just heard Meldon Sheldrake say something like there's 6 million spores airborne every year or something. So like spores and us interacting with fungi is like way more in depth than I think a lot of people understand, even in the food that they eat and the medicine that they take, you know.
Well, I love your philosophy because I'm with you. There's no such thing as a good guy or a bad guy. They all work together as a system. And if you have one out of balance, well, it doesn't matter whether it's a good guy or bad guy. It's going to change the ecology, and probably not for the better. It's just going to provide weakness. So yeah, before we go too deep on this, I'd love to talk a little bit more about your spent mushroom substrate. Are you refruiting after the initial, or do you just get rid of that because you're worried about contaminants coming?
I mean, usually, like now that I'm at a commercial scale, like in the beginning, I definitely try to get as many flushes as possible. But you know, I go for about 2, and then it's like taking up room that I need, you know, because I only have so many square feet to grow in. But we always throw them outside. I actually did this really cool project where I started lining my raised beds because I just used some wood with it, and you know, water wanted to flow out of that 6-inch drain, so I'd stack all the blocks around the side of it. And me personally, hunting mushrooms a bunch of times, people say pleurotus, which is your oyster species, has to, you know, it's a saprophytic mushroom, but I've seen it growing terrestrial in a grass field a couple times, and I've also seen it feeding off like minerals just right in this rocky runway of water, and it was like obviously digesting some really interesting things to be able to fruit. And so like I thought, well, okay, maybe if I just like stack enough of these blocks, you know, obviously we had a really great rain season this last time, and in our monsoon season it lasted from the end of June all the way to September this year. So you know, multiple flushes out of that, but I was trying to create like a terrestrial species. And so far, I could go outside and like show you a quick video that it's fruiting right now, and it was 8 degrees last night with snow on top of it. And it just goes to show the resilience of any organism in a certain condition. And so I'm always trying to do all kinds of stuff. My friend William Padilla Brown kind of had the idea of like, well, why don't we just stack mad blocks near these, shady sides of the building that get more moisture, and like try to recycle those. And I think those have, some environmental stressors that add to the enzymatic compound in the mushroom to make it a little highly more medicinal. And that's why I prefer, if I was going to make a medicinal compound out of mushroom, that I get some grown from nature, because I think, a set and setting, allows that plant to give me genetics from the area that will help me survive. And maybe I'm not, a biologist and I can't use the right terminology, but I think y'all understand what I'm trying to get at there.
Yeah. So I love the concept of introducing the substrate to the local environment. You ever heard of Dr. Efren Chavez?
No.
Okay, he's a guy I'm working with on a project down here, Wildlife Crossing. And we talked about doing IMOs and using different substrates instead of just the suggested undercooked al dente white rice. And one of the things that we both talked about was utilizing these substrates after they've been gone through. And because it is the perfect thing for collecting those airborne spores to drop in on and then go ahead and colonize. So I don't know. Have you ever thought about that? I mean, it sounds like that's what you're doing outside anyway. Are you getting indigenous?
And David, would you mind sharing some of the substrates that you do use?
Yeah. I mean, obviously in the beginning, I tried just about everything because I wanted to try to, pair with agricultural waste systems like coffee, or even like I knew furniture makers that were, throwing away their sawdust. But for them, I'm like, hey, separate the maple from the ass, and then that becomes a big chore for them. But I've grown on straw, I've grown on manure. I mean, a pretty standard growing medium in the industry is it's called master's mix, which is just like soy hulls and high pressure. It's just like a oak pellets for I can't think of the HFWP right now. I can't think of why that acronym slips my mind, but yeah, I've even recently been using like beet pulp and alfalfa, because down here they grow a lot of alfalfa, and so I know people that have used cannabis, but now there's more useful tech for cannabis waste. And so there's tons of different recipes. I like to pair with like spent brewer's grains as well, and you can kind of add that in the mix. And most of that information is pretty open source, either in mushroom groups on Facebook or other than that. And then Leighton, as far as like myco-remediation projects and stuff like that, I mean, there's so much potential there. There was like there's a lab down here that's been dumping toxic chromium waste in the waterway, right? And that water happens to feed into one of the biggest Navajo water basins down in the Ute Reservation up from Los Lunas. And so a lot of these farms are suffering, and there's a organization called Tiwa Women's United who was doing a thing and got the community together. And then again, Peter McCoy came down to do a myco-remediation project, and they had some pretty interesting fellows that were dealing with some of these radioactive materials come speak against it. And Peter found a way to get even dead mycelium. All right. So the dead mycelium, you could release it in the waterway, and the toxic chromium would attach itself to the mycelium for whatever reason, right? And it didn't even have to be living mycelium. And then that gave you the ability to now collect all this toxic chromium, grab this mycelium, and then recycle it. And now you're not even having to mine for the toxic chromium. So, I mean, the potential for like mycelium to do, I mean, it's, I've heard, they're grand molecular decomposers, I think was what, you guys talked about Paul Stamets, right? He calls them the grand molecular decomposers. There's a lot of research that shows that they can accumulate heavy metals. You can simply inoculate wood chip bags and put them in waterways and start doing, water reclamation projects and reclaiming, spillage from mine damage and all kinds of different implications. I mean, there's so many implications. So as far as like putting an open like block, I guess it depends on like for IMO, for example, maybe if there is a way that you could create a liquid culture at a big enough scale and like filter it out, and maybe if you like freeze dried the mycelium or somehow made it like a collection filter to where you didn't have the medium in between, the mycelium might be able to collect some of those like indigenous microorganisms in a better fashion than say putting it on rice, because growing with the rice, who knows what else you're getting that's coming from inside of that rice. I don't know if that's what you're getting at, but yeah, there might be a closed loop system that you can do that. Like I said, even the dead mycelium was able to attach itself to the toxic chromium and then recycle that out. So if you could do like, if you found like maybe like Ganoderma, which they use a lot in like the textile industry, right? Because they're finding that they can cook the mycelium and kind of like actually in a kiln and make these like bricks, and they're doing light fixtures, they're doing wine bottle instead of styrofoam, and so I'm sure if you found the right species of mushrooms and was able to isolate that mycelium and either like freeze dry it or cook it and then put it in a basket in your cedar collection box, you might be able to do something really cool. But I don't know, that was the first time I've ever thought of it. Sorry to go off a little bit.
Yeah, that's what we do here. We go off on rants. So I guess let me refine the question a little bit. Have you noticed in the areas that you've stacked up these spent mushroom substrates or SMSs, did you see any local indigenous mushrooms begin to flourish in it, or has it always been whatever you grew in it is coming back?
That's an interesting question. I feel like the living soil garden itself, maybe there was a little bit more mycorrhizal action, but again, just opening the lab and like having the microscope right there and, learning from Matt and all his amazing work that he's been doing with all this new R database, I can't wait to just really start looking in the microscope and inspecting those things. But it's only been like a year and a half since I've really intentionally done it. So I would say this season I will be more on the lookout. But in the last like 7 years, I've been at this property for 10 years. I'd say at the last 7 years, maybe it's just me going out in the woods and throwing like, random mushrooms around. But the amount of fungal activity is pretty good around the area. But I think what has happened is it's like, again, like I said, it's become like almost a terrestrial thing where instead of being a saprophytic thing, maybe it's feeding on the dead roots from the garden that's right there. Maybe the mycelium has somehow become like a mycorrhizal species where it's like now like that. I think the endophytes, maybe it's like you were talking, I think in one of your talks about like the glomulin that's like holding it all together. It's like, is this eating like this little tiny microscopic organic matter? And it's created such an intense system that it's like not even really eating a lot. Because I'm telling you, I looked today and there's a brand new fruit and it was 8 degrees yesterday here in New Mexico at 6,800 feet. And so, yeah, I don't know. I think anything's possible. I'll definitely keep more of an eye out and let you know when I know more.
Well, it's funny, but I don't think a lot of people understand that those blocks actually generate quite a bit of heat.
Oh.
As the mycorrhiza or as the mycology continues to grow and try to fruit and bind up all those materials in the block, it gets to 70, 75, 80 degrees in one of those blocks. So you stack a bunch of them together, that's going to generate enough heat to support a fruiting body even in a low temperature environment. So that's probably kind of explains why that happened. But look at what you've done. You know, by default, you've created a perfect environment for them to cultivate. And again, it's like, yeah, please keep an eye out, because it'd be really interesting that if you are starting to attract some of the locals, indigenous species of fungi to bring it to your bed, which is even better. I mean, that's just like a win-win.
Right. And then, being able to take those species of mushrooms that we like grew, and then to take them into the lab, and then to start putting them in fungal preservation and growing them out, and seeing if we can even use them in like horticulture applications, right? Like if this is like this some crazy terrestrial species that fruits in 8 degrees, like what do we have to feed it in order to, like you said, like create that system? Obviously it's going to take work in other places for people to mimic that, because it's like a seed. You get a seed from Georgia, it's great. I love collecting all these heirloom seeds, but as soon as I put them in the high mountain desert, I got this 8-pack of seeds, and I'm like, shit, you guys getting 8 in this motherfucker? And I put all 6 but 2, and then I might get 4 of them that work up here, just because set and setting. But luckily I'm like super into adapting things to their environment, improving the resilience. That's again part of my passion project, is like mapping the fungal evolution over time. And like, because fungi have so many different mating types, and I can't really grasp my head around this concept. I've asked a couple mycologists, and I don't know, it never really gets anywhere. So it makes me really want to like try to understand the concept of like how fungal mating types, and there's so many available. Like, does mapping the fungal tree of life kind of show the evolution of species over time, kind of like how an above ground fungus turns into a truffle and goes hypogeous? That's like an evolutionary trait. That's like, oh, you've reached it as a mushroom. Now I just have to go underground, and I can do all year round. I'm right at this humus layer that's perfectly ideal for me to live almost anywhere in the country, arid, semi-arid, drought, it doesn't matter, right? They find truffles in some of the most drought-stricken places. So it's like, that's that genetic evolutionary trait where it's like, well, yeah, I think a lot of these things, and there again, it's like, here I am trying to understand it, but there's this whole greater picture that I'm also forgetting to understand. And it's that's like what I was talking about with like mapping the mycorrhizal like networks, right? Like we think that these plants only have like a relationship with these host trees or not trees, but it's like, how can we even quantify how big this system that this organism is actually interacting with, right? Like how many other subspecies of mushrooms, like you said, are attracted to maybe like a pheromone or a terpene that this mycelium is putting out, and it's attracting all these other biological components in the soil, like the nematodes and all this other crazy stuff to this system, right? But we think, oh, well, like this is this mushroom, and it's always been association with this tree, so it's got to be this tree that's causing this mushroom to grow. Or is it like this one Armillaria species that we're calling parasitic, the Armillaria osteia, right? The humongous fungus in the Malheur National Forest, right? We were sitting here calling this thing a parasitic thing, although it's like one of the largest living organisms, and the ecosystem around it isn't dying. So like, is it parasitic, or is it like the police that's like going in there and like managing like, who's allowed to thrive in my ecosystem kind of thing? And maybe it's actually like mycorrhizal with some species of mushrooms together. They may, like, and then all these lichens and algae life forms, and like all this complex life comes together. And here we are saying, the honey mushroom is a parasitic mushroom that's killing this tree. Well, like, is it really killing that tree? I don't know. It's like the questions like that. So it's like, we got to even start to quantify and understand how deep these systems of mycorrhizal like mycelium are actually going and reaching, and like how to quantify that without an invasive technique is pretty hard up to this point, right? I'm going to tell you guys about this. I've just been coming across this theory the last couple of days. It just came to me, and it's like, well, how do we quantify this stuff, right? And so it's like, you could dig it up with a tractor and you can like go 20 feet down and say, yep, this my mycorrhizal network is going 20 feet down. Right now, we just destroyed that whole ecosystem. We can do what they've sort of done, right? And they can take a piece of this mycelium down here and they can go in this giant radius and they can start mapping it, and then they can start doing DNA testing, and then they can see, right? You know, maybe we could do something crazy like one of my friends who's in nuclear medicine, you know, I won't disclose any names or anything, but he did some really cool stuff where they were getting like radioactive material and they were attaching it to proteins, and those proteins would go into the body and attach themselves to the cancer cells, and they were able to map it, right? But then we're like, okay, well, we can't just inject radioactive dye into this mycelial network. Yeah, it'd be cool. We probably could see some shit and map some shit, but then we're probably going to destroy it, you know? And then my friend's like, yeah, that could be really invasive to that ecosystem. So I start thinking even more, and I'm like, okay, there's that I don't know if you guys have ever seen him, but he's like this conductor who went around in all these ecosystems and he started writing a piece of music for every species of mushrooms, right? So it started making me think like, oh yeah, these plants and these ecosystems are talking to each other, right? They're making these noises, and maybe our frequencies can't hear those frequencies, but they're alive, right? And so this guy's a composer, and I think he wrote like 74 pieces of music for each one a species of mushroom, right? And he hears the music when he goes out there. So this guy's dialed in. I think since now he's died, and since he started talking about that, people started quantifying, like they're hooking up machines to the mycelium, right? And they're actually like recording the sounds that these are putting out. And so I'm like, well, holy shit, you know what I mean? If these are like, you know, like, okay, how do we send a message to the aliens or whatever? Well, how they're sending messages to us right here. Now we're hearing it. Now we're measuring it. So like, how do we now send a message back and quantify all the way through it and like record that? And so like, I've had 3 kids, so I know that like, you know, sonar is a thing, and I know that ultrasonic, you know, ultrasound's a thing, right? And that's just sound. We're measuring sound and vibration. And so my one friend tells me, he's like, well, maybe like sending a crazy sound into this organism might not be good either. And I'm like, yeah, you're probably right, you know? So like, okay, a little bit further, I'm like, you hear about like the mother tree talking to her baby trees, right? Yeah, people definitely, I'll answer that in a second, but yeah, people definitely hook up these machines to the mycelium, and they're quantifying the sound that the mycelium is projecting, and there's a whole composer who did a lot of stuff, right? So I started thinking, okay, we don't want to send a sound into the mycelium. That probably wouldn't be good. But you think about like, well, these mycorrhizal relationships that they're sharing with these other organisms, right? And we've somehow quantified the fact that a mother tree will send resources to its baby tree before it sends it to these other trees, right? In the neighborhood, especially if it's one of its baby trees. Somehow they have this complex nature language that, you know, we'll probably never understand, but we hope to understand. And so it's like, okay, so how can I find 2 things that are living in symbiosis and symmetry in nature, right? Communicating obviously in one way, shape, or another, right? One's giving sugar, one's giving carbon, right? So how do we measure the sound that's coming out of that tree, possibly record that, send a message back into the mycelium at maybe like a certain hertz that's not really going to like change it? If, you know, again, there's no way to like know, but it would be really cool if we could send it back and it could reverberate, and through ultrasound and maybe LiDAR techniques, we could start mapping this underground mycorrhizal network and really understand and quantifying like what's going on underground. Like Av asked me, like, what's like, yeah, we see a lot of, you know, like I heard somebody say that the mushrooms don't want us to interact with them and they're elusive. Well, I think that's completely wrong. They're like, boom, they're bright red and they're all these colors and these shapes and these like enormous alien things because they want our attention and they want us to grab them and they want us to go with them, you know? And I'm not saying like we need to start mapping these, but I think if we could somehow quantify the complexity of what's actually happening in the system, we would have a better chance to be able to understand the bigger picture and what's actually happening, and stop labeling things like one way or another. Oh, this parasitic thing or this bad thing, right? And so it's like, I don't know, you know, those are just like my own personal theories that I've never really shared with maybe like 1 or 2 other people on the planet until the other day. But like, I think there's some really interesting stuff now with this LiDAR technique, right, where they're finding all these ancient civilizations, and they're finding food forests that have been in these jungles that were created, and they're able to map all this stuff, right? There's these kids on Google Earth that are finding these ancient civilizations by just like scrolling around, and it's like, yeah, like how do we incorporate that to map what's underground so we can understand and pair with nature a little bit. Sorry again to kind of woo.
All right, so
Baby, I love it.
So just to verify, yeah, there's been a tremendous amount of experiments being done where they're plugging in anodes and cathodes into mushroom, and it's playing music. And it depends on the species of mushroom, and trees and plants. They did on hemp plants. So they are generating a sound. So that's real. That's not sci-fi. And then I know people who have used radioactive isotopes, specifically Elaine, to map out how fast a tree releases exudates. So she was in a forest here in Oregon somewhere, and she had some fellow students down in the cave, like 200, 300 feet below the surface of the earth, connected to the roots that came through the cave. So they knew it was the same tree. And yeah, they popped in an isotope, and within minutes it showed up down in the roots, down in the cave. So we know these things are traveling very, very fastly. So there's a company that came out about 8 years ago, and this is kind of horrific, and I backed away from them. But there was two companies, one in Colorado, one on Long Island. Both were government funded, and they were making nanochips. The reason they were making these nanochips was so that once it's introduced to the plant, every single cell will have one as it's being reproduced. So it was a way to prevent counterfeit cannabis, or that was the guise that they were using. That if they introduced this nano chip or whatever to the plant at a young age, every bit of that plant material would have that chip. Now, there's a great way to do that, but again, you're playing with God, man, and that's where I kind of was like, that technology is too scary. You inject that into a baby, and now you'll have the DNA of that baby, whatever it touches, wherever it goes. It gets scary as shit. So again, I stopped working with them. I stepped back because the technology is too scary for me. And I again look at AI already, what it's doing. So those kinds of things are just going to start really dovetailing. And I don't want to get into control and conspiracy, but there are ways of doing this. And I don't know, David, if you've seen the recent information that was released 2021, where they actually had a microscope that was introduced in situ, so in place, and they were able to monitor mycelium over the course of a number of days. And what they showed, which was, this is groundbreaking, was that inside of that, mycelium network was actually hundreds, if not thousands, of different individual species, both saprobes and mycorrhizae. So we know now that they work in conjunction with each other. And we also know that the outer layer of the hyphae is very similar to a rhizosphere. There's an aqueous coating that's actually moving organisms, biology, down as like a super highway. Then it gets really fucking crazy. And they've indicated that these funguses, both saprobes and mycorrhizae, are allowing bacteria to enter into them. So like shedding its outer shell, so it's just the inner shell, like in an endophyte, is allowed to go inside of the mycorrhizae and be transported wherever. So, and saprobes too. So I mean, this is like crazy shit. That if you think about, oh well, what species is it? And you talked about the evolutionary. Well, it's gone to a truffle form now. And now you're talking about horizontal gene transfer, where two individual cells come together. And this cell got flown in on a bird and dropped in its poop, and now it's in a new environment and it doesn't know, how to survive as well as the local ones. And so they come together and they say, I got this tool. What kind of tools you got? And they trade. And that changes their ability to withstand changes in the environment, you name it, or drive an evolutionary chain. So I mean, I don't know if you've caught up on all that shit, but they're getting fucking crazy with this information.
Off anything?
Jeez. David, you're such a easy interviewer. You just, ask. You don't even have to ask a question, and you're gone. It's amazing. But I mean, I want to take it to a different tangent. So I'm not sure if Leighton's finished or not.
I'm good.
Yeah. Well, and I'm sure, David, with all your answers, you're incredibly holistic, right? So whatever question I ask, I'm sure it'll come back and we'll revisit a lot of these things. But I want to dive a little bit deeper into your commercial cultivation, and then perhaps even get into talking about some of your favorite species. It's just interesting because coming out of cannabis, where we're just always amazed at this cannabinoid can do this, and this cannabinoid, this newly found cannabinoid is doing this, and so on and so forth. And we go back, we often say that, yeah, we've been using cannabis as medicine for, so many thousands of years. Well, I think mushrooms predate all of this as well, right? And we're just scratching the surface of what some of these compounds are doing in mushrooms. So perhaps even sharing some of your ideas about what are some of the medicinal benefits that you're seeing in both gourmet or medicinal mushrooms, and perhaps I guess speaking a little bit more about some of your more favorite ones that you're growing right now.
Sure. Yeah. I mean, mycology is one of those things. Once you get into it, you're just like, I'm going to grow everything and do everything and find everything. So yeah, I've done a lot of experimenting. I think your question about like how long they've been around, I think cultivating plants is like 10 or 12,000 years, and cultivating mushrooms just over 2,000 years. But I think our intervention and interaction with them is like ancient, like goes back to like the archaic days, right? All the way to like panspermia, where they're like, the spore can land on the comet and hit Earth and like, essentially start to colonize Earth. I think NASA did a small project where they created an artificial meteor, inoculated it with different spores, released it in the vacuum of space, brought it back in, and they were able to scrape the spores and make them viable. I've said this a few times before, but that just goes to show that, yeah, I mean, the idea that a comet crashed with some spores, some mycorrhizal spores, they started mining the minerals on Earth, right? Started going at it and mining the minerals before there was any kind of plant life. We have to think like way, way back, right? And so in these estuaries where the freshwater and the saltwater meet, you have all this crazy organic life and just so much going on. And you have microorganisms and bacteria and like all these different things, right? And eventually those water's been here before any of it, maybe some minerals and some dirt before it was a soil, right? And then that maybe those building blocks came on that comet, and all of a sudden, right, now these mycorrhizals are meeting these algae and these lichens and these cyanobacterias and different things. Well, these cyanobacterias at that point, right? And then the fungi and the cyanobacteria meet and they become a lichen or an algae, right? Then we get these like non-vascular plants on Earth, probably. This is all just theory that I heard and put together in my own fashion, right? And so then we get these non-vascular things like liverworts and ferns and things start showing up, right? And we get this giant thing called prototaxides, if you haven't heard of it. Prototaxides is this gigantic mushroom things that have roamed the Earth at one point, and there's lots and lots of research. Since 2014 there was only one dissertation on it, but now they're like, is it algae, is it lichen, is is it mushroom? A lot of people lead towards like that combination symbiosis of this giant mushroom organism, right? And so like, yeah, we as humans have been interacting with them for a really, really, really long time. You look at Otzi or Otzi the Iceman, there's a whole thing about him, right? They're like, he's found in the Swiss Alps, down a couple mushrooms on him, Amadou mushroom or the fomitopsis. That's one of my favorite mushrooms. I've never cultivated or tried to cultivate it, but it just has a general amazing application to be a textile, to be a tinder conch, to be antiseptic, antifungal when you use it as a medicinal tea. So it's one of those holistic mushrooms, right? It doesn't just have its purpose. People are like, well, which mushroom is the most? I think they're all magic, right? So, you know, you look at, I have over here, I got the herba medica and the materia medica, like ancient Chinese medicine. A lot of these mushrooms have been in there for a long time, right? We just, in the Western world, we have the mycophobia. And so we get like, oh, you know, mushrooms are all going to kill us. We're all going to die. And not to go on too many tangents, right? But they find Otzi, they find this red lady, they find all these different people. The red lady of Miran, you can look her up in Spain. They found her buried in a super ceremonial way, and in her teeth they find agaricus, and then they find boletus. Usually people only eat boletes, you know, porcini, it's a very popular edible mushroom. So she was probably eating that for edibility, but agaricus, there's a lot of medicinal agaricus, so possibly she was using those. On Otzi, they found also the birch polypore. I can't remember the Latin name right now, but they use that for intestinal parasites, right? And there's other things like that. The tinder conch, the Amadou mushroom I was telling you about, you used to be given a task when in these nomadic tribes back in the day, and as the fire bearer, you would take the ember and you'd put it inside this mushroom, and the Amadou was real flammable, so you put it in there, you put it on your belt, and that was your job. You had to carry this ember to the next destination, right? And so beer, you look at all our novel compounds, right? Yeah, chaga's another really great one. There's tons of medicinal ones, right? But you look over time at our interaction and we think that like, oh, we understand them, but you know, they found fossil evidence in chitin in a fossil over 2 billion years old, right? So mushrooms have possibly been here for 2 billion, possibly since the dawn of time, right? Mining the minerals before the plants were able to make their jump out of the water and coexist with these fungal.
Oh, did we just lose him?
David?
Oh,
Yep.
I think David just froze up, guys. So you guys take over and I'll send him a text. Let him know.
Oops. Hey, come on, you come on. So that was a good question, Av. I like that. What was his favorite? He hadn't quite finished yet, so we'll have to, I guess, move on to something else. Is that something that you're working with up north, Av? Are you growing with mushrooms or teaching about them? You're on mute.
Yeah, I've got a couple of projects going on. One is you know, looking at lion's mane in particular, and looking at some of the neurodevelopment compounds within lion's mane, and just some preliminary trials so far have shown that in mice, just using eracine A or B, I can't remember which of the medicinal compounds out of lion's mane, but it's showing some remarkable regeneration of neural networks. So I don't know if are you consuming many of these medicinal mushrooms? But
you guys for a quick second there, the snow just came out of nowhere. But yeah, I heard the Erinacean question, but yeah, definitely Erinaceans. I try to take as many mushrooms as possible. My friend William had more of a theory that like eating fresh mushrooms is the best way to get the most medicinal compound, and I kind of took my own theories on that too, that like, you know, I'm not a biology expert and I don't understand, but you know, people say, oh, you got to have alcohol extraction, you got to have water extraction. And I don't know if anybody really knows, because a lot of the big research takes money, and then when you find a cure, they don't want anybody to have any of those. And so a lot of the research for this stuff is very, very new. But I definitely think that as a human, we're encoded biologically to process whatever good stuff this has, right? You might need to cook it and use some heat to break down some of the chitin to make it a little bit more bioavailable. But I think by just adding a little heat and making it bioavailable, our body can process those alcohol soluble and those water soluble compounds. Again, I'm not a scientist, so don't quote me on that, but I think we're pretty loaded with how to receive these medicinal compounds. And so, yeah, I mean, lion's mane, I'll go into some of my faves, is one of my favorites. But yeah, if you have something to say. Sorry.
Is it also a favorite to grow? Because it grows, in my opinion, relatively easily and so well.
Yeah, lion's mane, it might look like a harder mushroom. I say it's a beginner mushroom up there with behind oysters. Some might even say it's easier than oysters. They just grow so fast. They're prone to fruiting in a petri dish. They're prone to fruiting in vitro on a grain jar. It's really hard to keep up with the lion's mane mycelium. They really just want to grow. And if you find that mushroom in the wild on a tree, you could find a 10-pound specimen and take that home and eat for the whole week. So that's a pretty good find if you find a big mushroom like that. But yeah, these mushrooms contain such unique enzymes and compounds that are not found, I think, in sometimes in any other organisms on the planet, right? Like the Erinacea that you spoke of, like Hericium has Erinacea A and Erinacea B with tons of neuroregenerative properties, like you mentioned. But I don't think there's any other organism on the planet that produces that, right? And so you think about like the shiitake. I just heard it. I forget the name of the acid that it produces. It's ana something acid, and a lot of people think that that's responsible for the fifth flavor, the umami flavor, right? Is the acid that's the Augustyniak. I don't know the name of it. Somebody just told me today, but I mean, if you look at the shiitake itself, it's like producing, they say, all the beneficial amino acids, and it's the only organism on the planet to produce all those beneficial amino acids, right? I mean, cordyceps is producing cordycepin, which is pretty much an adrenaline analog, right? If you overlay the adrenaline analog, right? And so like going into these ophiocordyceps or cordyceps, they're doing crazy things for horticulture because they are an entopathogenic fungi. And entopathogenic fungi targets usually a host bug, right? And infects the endocrine, I think, in the endocrine system and another system in the bug's body, eventually mummifying it, taking it over, and usually fruiting a mushroom out of the head. Doing all kinds of different things, right? But it's like, so we have this invasive red lace moth come into the East Coast a few years ago, maybe 5 years ago, right? It came in, the whole East Coast started destroying all these grapevines, right? And here's the FDA, and they say, oh, we got this phallic solution that you can spray, and it's $300 a tree. It might kill the entire ecology in the soil. It might kill all the microorganisms in this entire environment, and your tree might survive. And it's $300 per tree. And if you have like hundreds of acres, like how are you supposed to apply this to your system, right? Where there's like a entopathogenic fungi that shows up in one of these mycologists' farm that I know, right? And now he's able to take that, culture it on a petri dish. He can infect a host bug. And this is a host specific, right? It's a parasitic response to a parasite. Dare we use the word again, right? But it's like this parasite comes in and nature's response is to create a parasite to attack that. And host specific, safe enough to spray in your own mouth, and you infect these host bugs, they go back to the colony and everything's disrupted, right? So like these enzymes and these compounds that are found in these fungi are like just amazing things that we have so little light on. I think the neurologist who discovered the potential with the lion's mane was barely in the 90s, you know, and not to say that we don't have the information. Maybe it's just being like given back to us, or was hidden for a while, and the mushrooms were kind of just like, no, we've been here, we're gonna keep being here, right? And it's like the overall mega science that we barely are starting to understand, because there's so many other sciences that really could relate to it. Again, it's that whole system of everything coming together, right? It's like another thing I'm going to tell you guys is a really crazy philosophy of mine. Maybe somebody's out there doing it, maybe somebody's not doing it, maybe you guys can lead me in the direction of somebody who is doing it and like get some really cool stuff going with them. And so like I've been having these really interesting theories. Like my really good friend Julian and another person talked to me about it today, is like terpenes, right? Like we've been doing a lot of research in terpenes since the cannabis industry came up. And a terpene, as far as I understand, is an aromatic compound produced by some sort of organism. I think we've always considered it plant organism up to this point, right? And so like I'm like, oh, okay, right, you know, I'm trying to understand what a terpene is. And then like I thought for a long time terpenes only came from cannabis, and then I'm like, oh, okay, no, you know, terpenes come from limonene, and there's all these different ones, beta caryopylene, and all these different ones. And so I'm like, okay, if these other organisms are producing terpenes, why aren't we like looking at mushrooms and fungi for terpenes? These are some like, my olfactory is a very sensitive thing. It's one of my wonderful portals that I get to use. Sometimes I didn't have the best relationship with it, but now I'm on a really great relationship. You look at a truffle, it's one of the most aromatic compounds on the planet, right? Its job is to literally create aromatic compounds to get other organisms to come digest it and spread its biology throughout the forest, right? It's like, why are we not looking at these? And I observed it, right? My second nature when I'm out mushroom hunting, besides pick it up and look at it, is to smell it, right? And these mushrooms are producing some of the most intense aromatic compounds that I've ever smelled in my entire life. A Foliota species, right? For example, it's this really crazy garlic smell. And like some people don't smell it, some people do smell it. There's these little polypores that are producing this like lavender smell. It's like a lavender cookie that I just want to live in. One of the best smells ever. And it's like, why aren't we trying to measure and quantify the terpenes in these things? And then I start talking to my friends, just trying to be a little secretive about it, try not to spread the information too far, because I want to like do some research and get somewhere with it. But like, fuck that, right? Like, why? Everybody needs to be doing this. If you have the resources to do this, do it. Start doing it. Share with me. Let me know what you got going on, because I think that like, you know, the entourage effect is what I just learned about, right? It's like, if you guys don't know, I'm sure both of you know the entourage effect, right? But it's like using terpenes with cannabinoids to create a super awesome effect. And my friend Julian really turned me on to it, but this other guy really made it a simple way to understand. So I've been a cannabis user my entire life, since 13 years old. And yeah, I love cannabis, but you know, like cannabis is like the bus, right? It's driving us to where we go. And these terpenes are like the side road, man. They're the side road to get you to your destination, right? You can ride that bus all day long, but if you can get on these side roads and really reach what destination you're trying to do and pair with it, you can have some really amazing effects, not only in like your own mind and what you're trying to do for your own temple, but like we could put these applications into the medical field, right? It's like I heard from Ken earlier. Oh, it's the hidden elm. Thanks, Julian. That's my buddy. He's tuning in. We do a lot of cool work together. He's helping me organize the festival, right? But hidden elm is this really amazing smelling one, right? And so I even observed it in my own growing room as soon as I recently just trained substrates, right? It's like I've been growing oysters for a really long time. And they smell, and each species has its own kind of smell to it, right? But I changed the substrate up to where it was like growing with this alfalfa and these beet pellets. And I was in the grow room, and maybe it was because of the cold and the environment. And then it was like just really excreting some really intense smells, and I'm gonna call them terpenes. You know, don't quote me, but I'm gonna call them. This fucking thing was pushing out some interesting terpenes that I had never smelled from it because I'd never grown with that substrate. And then all of a sudden, I'm like, really smell it. And I get the guy who's working with me in the biology and the lab and stuff, and I'm like, Mac, do you smell this? And he's like, well, I smell something. And then I'm like, do you smell alfalfa? And he's like, whoa, I smell alfalfa like fuck. And then I'm like, yo, so can we start pairing with these organisms and feeding them certain substrates to get different like terpene content? And like now how does like environmental, like obviously in the cannabis industry, all those things play a very important role in the terpene, right? When the weed turns purple, we get different terpenes and stuff. So it's like, how are we not applying this to the mushrooms, right? I don't know if you've ever smelled shiitake mushrooms, but they got a certain smell to them, right? And it's like stink horns. Like, what the fuck? One of the stinkiest things you could probably smell on this planet. If you've never smelled one, whoo, good luck, right? Some of these, like truffles, like I said, you know, and so it's like, I really want to start like isolating some of these terpenes in the mushrooms, and then creating some entourage effect in the medicinal world, talking about like how I want to apply it medicinally. That's like one of the effects I have, you know. I think it's the full body medicine. Like how do we isolate some of those terpenes and then eat them with our lion's mane to have like the most restful sleep and neuro regenerative REM sleep of our life, by like isolating part of this reishi that's going to allow like awesome blood flow, but we like get some of that terpene, then we stack it with the lion's mane mushroom that we ate for dinner, and then all of a sudden we're like in perfect REM sleep for 5 hours, and then we get up and we're functioning, you know. It's like, I think that this would be like something that more people need to. So if you want to like team up with me, I'm just like getting this citizen science lab going, and like anybody in the cannabis industry that has a way to like quantify and register these, or can like point me in the direction of a lab, because, you know, writing with a lab and doing a grant and like that's all great, and I have the resources, but that's going to take me a shitload of time. If there's like somebody doing it right now in the cannabis industry for other shit, like, yo, call me up, I'll send you all kinds of mushrooms right now fresh, and we'll start like doing some really cool shit. And like I think everybody should be doing it, and that's why I wanted to talk about it today, because I think I did a little bit of back end research and like looked through all the publications and stuff, and there's like very, very little research. They've identified some terpenes in mushrooms, but the research is not there yet. So anyway.
All right, we're going to take a 33-second break, guys, and we're going to show you who's coming up next week. So, Dave, if you got to go for a quick pee, go ahead. We got 33 seconds.
All right, we're back. Yeah, you know, I think I think you're correct, David, that they should be classified as terpenes, but you can always say volatile organic compound, VOC, because that's another way, another expression for odor that comes off as a result of oxidization or exposure to sunlight. And yeah, in a lot of ways, medicinal, you know, especially you know, Eastern medicine, they've been using, you know, aromatherapy for thousands of years. You know, so you're spot on what you're talking about. Ab, did you want to add something to that?
No, I was, you know, I had all these questions around the entourage effect and the fact that, you know, David's all about a whole body medicine, you know, a whole natural extract, full spectrum extract, which is, and it's because I was thinking that you wanted those terpenes, right? The terpenes that are in fungi, and you wanted those with whatever other medicine was in that. And so I thought that's what we're thinking. And so I think that was bang on. I'm a big fan of thinking that way in terms of you know, not trying to isolate a particular molecule out of these mushrooms, but allow it, as you said, like a citizen scientist, but citizen medicine as well. So what we can be able to cultivate our own medicine from these natural plants and fungi stuff. So I thought that was, you know, perfect, phenomenal with that. And yeah, terpenes, the way I look at them is I think it's I read somewhere it's the most common language on Earth, right? Or it's a most popular language on Earth because bacteria use that to communicate with plants, with fungi, with other bacteria, and I imagine other microbes in the soil are using terpenes as their communication tool. So
I think they call it chemotaxis, where one smells the other. There's some really interesting stuff done on how a spore, whether it's mycorrhizae or saprophytic, can find its host or find where it wants to go. Where's the source of my lignin, or where's the source of my host plant? And they use that, the plant's excreting an odor or a smell, a chemo, and a fungi is following it. And I mean, because think about like a spore hatching or coming out, sending out a shmoo in the soil, 3-dimensionally, you don't know where the hell you are. But it's nature's proven that it can find the direction it needs to go, and have enough energy. Like it won't release, it won't come out of that spore until it knows it'll have enough energy to reach the next source. So yeah, really, really cool stuff on the understanding of chemical signaling and electrical signaling too. And Ab, this was a question I was dying to ask you. Have you played around with exposing the cannabis plant? Say you have a cherry pie cultivar, and then using cherry tree shavings and leaves to encourage and get a greater expression of the cherry terpene and flavors.
Yeah, I think that's something that I hear Kyle Cushman did, right? Trying to get some more strawberry flavor and strawberry cough, or more blueberry into blueberry dream. I'm not specific about that. No, I've never played around with using. I was interested about the whole alfalfa. I've never been used that direction, but definitely using microbes to enhance terpene synthesis, right? Or a particular. We did play a little bit with jasmonic acid to try to increase responses through the jasmonic acid pathway to increase cannabinoid synthesis, but that's about as much playing around I've done with that. But I would see that it does make sense if those bacteria give strawberries some of their flavor in that terpene production. Why not increase that? And so by adding those bacteria that you might get from a strawberry field to that area, or that might be present within the strawberry. And I think when we think about UK cheese, right? UK cheese, it's a one particular terpene that kind of does. It's a very non-dominant terpene. So if you did a terpene profile on UK cheese, it's like very low down within that terpene profile that actually has the strongest cheese flavor, right? I would be interested. I mean, it's a great experiment. Use more of that bacteria to see if you get a cheesier flavor.
That's what's up.
I think that's what you experienced, David, recently, was that the mushroom had pulled up, whether it's the chemistry, the biology, or whatever, out of your substrate and released it when you were present in the room. So again, this is like just another example of taking advantage of nature's own processes to better enhance what we're looking to do. So keep playing with that shit, David. By all means, man.
But what's interesting with fungi is that because it's such a great bioremediator, you can put it on substances or grown in substances that are perhaps toxic, and yet the spore or the fruit does not contain any of that toxicity, right?
Well, so me and my friend William were talking about this a couple years ago, and I think that, like, I don't know, it's hard to say. So it's like, you grow a cannabis plant and you clone it, and then you clone it, and eventually it starts to lose its like genetic diversity. So some people say, well, when you keep cloning, you make it a more stable clone, but I think that depends on like if you're growing indoor. I think there's a whole bunch of different factors to it, right? And so as far as like the mushroom goes, I was trying to like do these projects outside and like grow these really drought resistant, cold resistant strains, and then put them on agar, and then try to put them in the fruiting room. But I noticed a lot with psilocybe. Psilocybe have this really good ability to come up with like a cure for any kind of bacteria that's in there. And I feel like in those enzymes, there's a lot of information, and that mushroom's creating that enzyme to feed it to itself so it can create a resistance. And so me and my friend got in this argument about like, okay, well, I'm going to create these really harsh environments, and then I'm going to clone the tissue afterwards, and I'm going to put that on agar, and it's going to have. And he said, well, no, the biology is not going to be there. You're going to want to collect the spores, and then you're want to grow the spores out, and then you're going to have the genetic diversity in the spore. But like growing with so many different plants and cactus and different organisms, I think environmental stressors can definitely change the genetic makeup of an organism. In my own personal experience, especially like with cactus, maybe not as much in the plant world that I've noticed, but I think very much so with mushrooms. I think mushrooms are definitely communicating to themselves in a way. And plants, I think everything has its own ability to do it. But it's like, I grow a lot of Trichocereus cactus, and the clone comes from the mother, but it's more like a tree relationship, right? Like where that clone comes with the ability from its mother, like the mother's like, here, I'm passing you all the stress I've had for the last 20 years in this environment, and you're going to grow knowing encoded. And that's just a clone, right? That didn't have to come from the seed. And I feel very strongly about that with cactus, that environmentals, because I've made high mountains South American cactus live in a greenhouse where right now I'm looking at like 3 inches of snow that just fell since we've been on this talk outside, and they're in a greenhouse by 2 pieces of plastic and a very small heater that goes down to 32 degrees. Now that's not ideal for those cactus, and maybe I've lost a couple hundred that I didn't want to lose in experimenting that I've grown from seed and not from seed. And it's not growing from seed. Even, we'll find out this year because I'll fully close the cycle, because I collected seeds of some that grew, that flowered in this environment, and I'll seed those, and then I'll grow those and see where they play along the spectrum. But I feel like mushrooms are kind of the same as the cactus, right? They're getting that environmental stressor genetically encoded in them for them to survive, because you can clone it and clone it and clone it. And I've seen it a lot in psilocybe growing and like the diversity. A lot of people like create these great canopies in their grows and stuff, but there's no genetic diversity, right? And for me, the magic of mushroom is like picking the small mushroom or the big mushroom, and that small mushroom's got packing more than that big motherfucker, right? It's like cannabis percentage testing to me. They're like, well, we tested 2 buds on 15 of these plants, and it's a consistent 70% or 20%. And you're like, no, motherfucker, the top to the bottom is going to have like a range of at least 10 to 15%, you would think, if you like had a little bit of a logical approach is like what's happening in the plant. And so I don't think that seeds are the only way. I don't think the spores have like all of the answer, but I think like if I wanted that terrestrial species to grow again, I need it to sporulate in the environment and drop spores in the environment, and then work with the saprobe mycorrhizal double relationship like you were talking about that they're encoded with already. All those local fungi might have been waiting for a saprobe to come in there and activate it and be like, boom, here we are, now we're going to grow. Yeah, I'm interested in collecting the spores and like seeing what the potential of the spores has to do, but I'm also collecting that mycelium to see how it's like genetically adapted to its environment and survived, and bringing it back into the fruiting room. I'm interested in creating contamination in certain projects, not in my whole fruiting room, but in some of my fruiting projects, so I can see how the mycelium behaves when there is like a viral infection that comes in, and then can I isolate that enzyme, take that that enzyme, introduce it into the water that I'm hydrating my bag with. Now I'm genetically coating the mycelium with an enzyme that's like giving it a precursor to digest or build up, we won't say like a resistance, but we'll say like a resilience to some of these biological factors that we experience in a grow room, whether it's clean or not, right? Like, how can we introduce those into that mycelium and then train that mycelium? It's like, because I've seen it where they trained mycelium to eat cigarette butts, right? And they didn't use new spores every time. They used the same mycelium, and they started reintroducing the plastics on the petri dish a little bit more at a time, until eventually they just took the whole entire oyster mycelium that was on that petri dish and filled the whole sterilized bucket of cigarette butts. Peter McCoy did this with one of his buddies, right? And so, like, yeah, environmental, I think, plays a huge part in it, and it doesn't always have to be passed on, but I think the most answers are going to be coded in the next like genetic sequence, or however that works. But I definitely think the mycelium, you could do a lot. And now that I have like access to this lab, like, you got to understand, a lot of my approach to this was doing this low-tech open source. So I grew a mushroom farm for 3 years with no lab, no still air box, no open flow hood, none of that. I did it all in my kitchen, the dirtiest place in my house as far as like biology goes, right? A little bit of spray alcohol, a clean shower, and like good aseptic techniques, and I was able to do, you know, hundreds of pounds a week on my kitchen counter, or on a table, not on my kitchen counter. It was on a table that I designated for it, you know? And so, yeah, there's just so much that we can observe. And like you said, Leighton, just like observing nature in these systems, in our down blink of humanity that we get, if we're lucky to get 80, 100 years out of it. But yeah, I think, the answers are definitely all there if we're willing to just like tune in a little bit. It takes a little bit of extra focus, but there's so many different things happening that I wish I could answer all the questions for myself. But now I got time a little bit more. I got a little lab. I got getting some resources to do some really fun, cool stuff, and learning more from experts like you. And now I'm just gonna like follow both of your work forever, of course. And so it's like, it's great to be on today. And somebody had a question about IMOs. I'll do my best. Oh, okay, yeah. I would say reach out to my friend Ecological Projects on Instagram about that IMO KNF style. He's, when we met, he was already a fermenting master, and I was like, hey, have you ever heard of KNF? And he, of course, like knew everything about it, but didn't, and then now he's like the wizard. He does it all. He's the fermenting guru, not just KNF, but he does Jadam and natural farming, and I'm lucky to have him around. He pushes me and inspires me to do a lot of really cool stuff. And I got to go to see him talk about his Jadam cool stuff. So yeah, I mean, we're definitely going to try to build some boxes, and at our festival, he's going to do a bunch of coffee, and we're going to inoculate the waterways. And like, as far as IMOs, I'm not an expert. I know very, very little bit about natural farming. Of course, I think it'll all fold into itself eventually, but there's definitely reach out to him, and he can give you some good advice. So, yep.
So here's another one. Been doing a lot of work with SMS, spent mushroom substrate, coming out of LA, and utilizing it in making compost to get a better F to B ratio for my clients and my own use. And interestingly enough, when the bags come in, there's often a yellowy liquid underneath them. Have you had any experience with that type of leakage?
So that's actually, the metabolite or the enzyme I'm talking about, right? And so like coded in there is like, right, I don't think anybody's touched it. The first person I ever heard talk about it was my buddy Alex Dorr. And I think I've also heard somebody else talk about it, Trad Cotter. But yeah, those are enzymes, man. Those are like, there is some crazy biology going on right there. And like I said, it's been in this grow room, in this environment, and that's like, hey, I'm running out of food, I'm stressed out, I'm going to create this metabolite to try to keep myself alive a little bit longer. And then it's like, produce this food. I don't really know, to be honest, but yeah, those are metabolites. Those are those enzymes I'm talking about that I'm observing when I'm growing, like, the psilocybes, and I'm seeing these viral things come in, and like mushrooms have this really intense, even I think Trad Cotter was putting 2 different myceliums next to each other on purpose on a petri dish just to see what would happen at that cell wall. And they would create these really thick cell walls and then build up enzymes to like kind of create this fungal battle between each other, right? And he even went as far as to grow specific, this is Trad Cotter, specific medicinal mushrooms, and he created this special bag, right? And so he started ordering like H1N1 and all these different viruses, yeast infections, and all these things. And you could actually fully colonize the bag, right? And then you could take one of these viral infections, you could wipe it on the mycelium. And again, like I just described, that mycelium is going to build this really crazy cell wall, and then it's going to produce an enzyme. Now you isolate that enzyme, and you can take that enzyme, and now perhaps you have an antibiotic that's specific to H1N1 virus, right? And so the potential for that could be, we have these blocks on storage at the doctor's office. You go in with whatever infection you have in your throat, you get a throat swab, they wipe it on there, and now it's like creating a DNA specific antibody for you, just by these medicinal mushrooms. And he started testing it with all kinds of different medicinal mushrooms. Same guy who was doing the entopathogenic fungi, where he was creating like pesticides that are safe enough to spray in your mouth that are targeting specific host bugs in these horticulture systems. And yeah, I mean, mushrooms, again, just like, yo, let's keep pushing it. But I don't know if I answered your question at all there.
Well, you did. I mean, you validated my own findings. It was explained to me that was a biocide. So when that fungus is being threatened or not in the perfect environment, it excretes that enzyme because it's in a weakened state, to protect itself from something else coming in and taking it over. And in my experience in the work I did, I took some of that liquid and introduced it to a very advanced biological compost to see if I did get a reduction, and I definitely did. So it's really interesting, because you're absolutely right as far as future medicine is concerned. To put these group of mushrooms together and have them build that wall with that biocide in between all of them, you're going to get a more diverse biocide that would be tailored to whatever it is that you're looking, based on the genetics of each one of those cultivars, to get a certain antiviral or antibacterial biocide. Takes everything the fuck out.
Yeah.
So really cool.
Yeah, so I there's probably a ton of research to be done there too, like, with soil biology and things like that, and how do we introduce certain pathogens, or like even like it could be an above ground pathogen, right? That's like killing a crop, and say we introduce it at like a mycorrhizal level, and now it's producing this enzyme that we could take and replicate it at a large scale and start spraying it as a foliar, and now this plant is building up a resistance to that pathogen or that disease that's attacking it. Perhaps, I don't know, that just came out of me. I don't know where that came from, but I think that could be an implication that somebody should jump on.
Absolutely. Ab, you wanted to add something on that one?
Yeah, were you both talking about like quorum sensing? Is that?
Well, that was where it goes. But what we were talking was actually about the excretion, this accumulation of an enzyme, a peptide. Who knows what it really is? It to be determined. But it definitely, when these fungi become stressed out, whether it's environmental issues or they're becoming weak and they're running out of food, they go into this defensive mode where they excrete all of this biocide. And next time I get, oh, that's right, we don't get the mushrooms with the blocks with the bags on them anymore. I was going to say next time I get one, I'll shoot you a picture of what it looks like. But it got to the point where I told the guys at the yard, I want you to run over every single block, bust the plastic so the biocide comes out, because I don't want to put that biocide into my biologically diverse compost, because I'm going the wrong direction. I just want the spores that are coming out of the blocks.
Interesting.
So, yeah, it's really interesting stuff. But yes, that's where it's going to lead for sure.
Just jumping around again. If you don't have a question, Leighton, I've got plenty for David here. So
yeah, go. Trying to be fair.
This one actually goes back to comments you made in a conversation we had around mycorrhizae in particular, and the fact that ones that you can commercially find viable or commercially available tend not to be overly viable. And I think you I hope I'm not publicly putting you in a bad spot, but you said that you did find one out there that you really did like. David, have you had any success generating mycorrhizae and propagating them to in a way of being able to inoculate other species on your own farm by any method of propagating them?
Yeah, I think, I heard something, I don't know if the terminology is right. Maybe they called it like rewilding or like wilding your property. Like when I first moved here 10 years ago, I was mower. Like I mowed and I did tilling and I did all that stuff, right? Because that's how traditionally I was taught to think. And then I heard somebody say, if you leave the biology undisturbed for 5 to 7 years, the landscape will start to renaturalize, I suppose, or go to its like native. It's like when you drive out here in New Mexico, there's lots of open fields, and if you drive for a while that hasn't been touched by anything, it's like the pristine nature, and you can just tell nature's doing its thing, right? And so I try not to like mow and do any of that, right? And try to increase as much of that. But, there's interactions. I build things, I put greenhouses, and it's really funny to see the successions of quote unquote weeds or reparative mechanisms, we should start quoting them as, right? It's like, again, it's like, oh, that human intervention, this is the succession that's going to grow, and it's going to interact in this manner and in this way. And so I had a friend, my buddy William Wildcat, he had taught a mycorrhizal similar inoculation thing, and a great way to do that is to just go, if you don't have property that is fruiting mushrooms abundantly, at least go in your local ecosystem, collect a bunch of mushrooms. This is the only time I would say collect as many different mushrooms as you can think of, and not do something as far as like science or like preservation or trying to ID them. Everybody does it, don't get me wrong. I was guilty of like, oh, I'm gonna go ID 60 mushrooms today, and like doesn't happen. But in this case, especially if you're in a good rainy season in the mushrooms, go out, pick 60 30 different species, take them all home, dry them out, try to get more mature specimens that have sporulated, dry them out, and now you have billions and billions and billions of spores of native mycorrhizal species, right? You put them in a 5-gallon bucket, you crush all that up, all that biology up, you fill it with water and you mix it around, and now the spore dispersal, wherever you're going, you could probably do a topical foliar spray across the whole property. For me, I try to like think, okay, you can break it up even better. You can go and say, hey, this is all growing with this ecology. I'm going to harvest these, and I'm going to take them to this part of this restoration project or this property that I want to increase indigenous mycorrhizals. And, you put all those together and you spray them around those trees, you squirt them around. That's a lot of spore dispersal, right? And like, there's the 5 fungal needs. It's like the right substrate, it needs water, it needs fresh air, it needs a good food source, the right food source. and I always forget the fifth one every single time. But either way, right? Like all that spore has to do is land in that 5 condition. And like, you know, these mushrooms, like I said, that gill has like, down that gill is tons and tons of basidia, and on every single basidia there's 3 spores. So there's millions and millions and millions of spores. And you know, mushrooms are good at making lots of them, and only maybe one lands in the ideal condition. You know, you talked about like the ability for those spores to stay in the mycelium itself. Like, well, somebody told me about like they were observing spores in the ocean, and these fungal spores developed their own flagella and started swimming around the ocean, man. Like a spore developed its own flagella and just started paddling around the ocean looking for compatibility. Like, talk about like crazy shit that they have the ability to do. And so yeah, it's crazy. Mushrooms are awesome.
Leighton, that kind of halfway sounds like what you were doing with your reclamation project for Roundup and that, where you would get the worst of the worst and then, you know, propagate that in, you know, for reclamation projects.
Yeah, definitely. It's similar. I'd never heard of a spore growing a flagellum, so that's really interesting. Are you sure it wasn't an algae that because algae tend to be mobile and they can look like spores, similar to like a flagellate, but that's wild, dude.
I would say reach out to Alex Dorr of Mushroom Revival. He was the one. I don't mean to like get him on a spoiler, or maybe that was some like crazy research I wasn't supposed to share, but like, yeah, he told me about it a few years and tell you right. It's been about 3 years, so I think he's probably come with whatever he was going to do with it. But he told me, so I think it's fair to tell you guys about it. But I think it was a pretty cool mechanism. That it was definitely a fungal spore. He's a pretty up there mycologist. So I think he was onto something. It's just a weird thing, you know. They're now finding fungi in like ocean and waterways, and you know.
Oh, there's a tremendous amount of aquatic fungi, a tremendous amount. Fishbrew.com has I don't know how many different cultivars of aquatic fungi, and you know, you start listening to not David White. I have a Dr. David White here, Dr. James White, who does the endophytic biology, and he'll talk about how the endophytic fungi will come out of the leaf and turn into a saprophytic fungi until that it's reintroduced to a new plant. And when that dead leaf falls into water, that fungus actually becomes an aquatic fungi. And in many ways, I truly believe that both the terrestrial and the aquatic environments are interactive, are part of the same system, and that the biology can adjust as necessary. Again, remember they morph. I mean, they're quick life cycles, so they can change over the few generations, make huge modifications. So yeah, and another friend of mine is actually doing something similar to what you talked about, using spent mushroom substrate in these giant cages and sinking them into these pools from a lead mine to try to clean up the lead that's coming out of the water or coming out of the old mine, and having tremendous successes with it. He also did some work at a university where he introduced white rot fungi to atrazine. Atrazine's a horrible pesticide. Has a very, very long half-life. And they were actually able to, just with the fungi, break that atrazine into secondary and tertiary metabolites. The only problem was those metabolites by themselves, yes, they've reduced the half-life, but they're still horribly toxic. So he called me and asked me to send him some of my product. And when they used both the white rot and with the highly intensive biology, they were able to eliminate all of it in a very short period of time. So we know for sure that in combination, fungi and highly advanced biologicals will pretty much do anything. as far as breaking compounds down. Now, that hasn't been tried with the forever chemicals, but I know it's been tried with, I've done it with hydrocarbons, did it with atrazine, did it with Roundup. So we know that they will work. It's just a matter of, again, finding the right constituents to do the work, or shotgun it, put as many different diverse organisms as you possibly can, and let the one that's going to do it. But in the Roundup case, yeah, no, I went for specifically a horribly exposed fields, corn and beans, where they use just so much herbicide there. It's not funny. And I told Kyle to dig up some grass clods that had been exposed to this in a field that he knew was extremely dirty. And I took the grass clods and I separated the dirt from the root ball. The root ball went into the compost pile. Then I separated the liquid from the sand, silt, and clay. So I had just a liquid, applied that to the area that had been treated pretty drastically with Roundup. And now if you go outside, you can see the garden is thriving, whereas it was all dead. It was just earth when I got back. So yeah, there's a lot of ways to use this stuff. And again, if you're using fungi to mine like chromium, and then now you can use the chromium that you got the fungi to pull out of the toxic place, and you can use that to refine it and make more chromium for whatever you needs. And so, and there's another woman down in Mexico doing this, using fungi to mine specific minerals that are both rare and very hard to get to, and then using the fungi to break the fungi down, and so that she got the minerals without exposing workers to horrible conditions, extreme heat, toxic air. They all got to be gassed up, and it's crazy. So the future is wide open, dude. Wide open. All right. Anybody want to add anything? David,
you're on mute, David.
I was just gonna say, to fully close the loop, yeah, just go out and collect those actual mushrooms, because they do have that spore that you can spread around your site, and try to work with native stuff, if you can, so you're not. It's really hard to understand what's really going on in these complex systems. But as far as like concentrating, definitely dig up native grasses, and your chances of isolating the mycorrhizals in those. And then I haven't had a lot of experience with it, but I know cultivating native mycorrhizals isn't super hard if you can get the right grasses that are, pairing with them. You can always go to your local museum of biology, and they could probably tell you the endophytes that are pairing with certain grasses that you're trying to target. And then you can go to the museum. They'll literally show you where that grass is. It could tell you a site. You can go harvest some of those mycorrhizals, put them in different containers with that native grass, and now you can feed it the right biology and increase production of it, and then you can harvest the mycorrhizals there. For as far as like cannabis production for your own personal use, I definitely heard Leighton talking about a lot of that biology. That's the whole spectrum, right? That's going to grow the best plant. I've been into living soil for a while myself, so I definitely think your local environment is going to have the best ability to give you what you need to grow there the best, I think. And so, yeah, it's pretty replicable system. There's a little bit more technology. Again, look to Peter McCoy's book, Radical Mycology, has some arbuscular and all the EM and all that cultivation and how they're doing that stuff. So, yeah. Seeing lots of puffball mushrooms that have Berlin mushroom and spores, and if I put them in my worm bin, would that be a bad thing? Nah, puffballs are pretty much just a giant. I don't know if the mushrooms or if If maybe inoculated afterwards, that would probably be a question better for Leighton, as far as like the biology of how to inoculate your worm compost would be. I mean, if you're feeding the puffballs to your worms, I don't think it'd be a bad thing. A great thing is spent mushroom blocks. If you do have a mushroom farmer in the area, mushroom, again, it's producing that compound all the way at the mycelial level. It's super sweet smelling a lot of the time. Different species of mushrooms have their own aroma at all levels, but worms are really attracted to that sweet smell, and they like to eat the mycelium, so.
No, I don't think you'd have a problem with the puffballs. I mean, as long as you want to grow mushrooms in your worm bin, because that's what's going to happen.
That's what's going to happen.
Cold, dark, full of fucking food. Yeah, pretty much, and water. It's a pretty much ideal place for mushrooms to grow.
And the puffball is essentially just a giant spore ball. So
yeah. And what's funny is Efren freaks out every time I find a puffball, and I don't know why. I think that puffballs may be more mycorrhizae dominant in as a whole. Like if they're going to produce X amount of puffballs versus actual mushrooms, I think that they generally produce more puffballs than they do mushrooms, whereas the saprobes are way more mushroom and very little puffballs. At least that's what I've been told. I don't know if that's fact or not. But yeah, so if you got puffballs, that's a good thing. Ken, we're getting up there on time. Is there any other questions that were highlighted that you want to put forward?
Actually, I've been trying to bring them up as people have been asking them. But if anybody's got any questions, just throw them in the chat. Yeah, we got 20 minutes left. So if there's questions, guys, I'll bring them up.
Yeah, was there something else you wanted to hit on, my friend? You said you had a bunch of questions.
Jeez, where am I at now? Turkey tail.
Oh, sure.
Got any good tips on turkey tail?
I mean, a highly wide distributed mushroom. Where are you at in the world? In Canada too?
Yeah, in Nova Scotia on the eastern coast.
Oh, I'm sure that they're available. They're a super good winter mushroom too. They come out late in the fall. They come out early spring.
I was actually thinking of commercial cultivation of them. Have you tried doing any cultivation of those? Yeah, could we see the false turkey tail here at times, and the turkey tail.
It's such a like widely available in the outdoors. I personally won't say that you shouldn't cultivate medicinal mushrooms, but again, it goes down to like my theory about like with psilocybe. So for example, like there's probably some psilocybe growers out there that are like, yeah, and so like we're cloning this tissue, right? And then we're recreating this mushroom over and over and over again, and they call it a canopy. So there's like hundreds, but it's the same exact mushroom over and over and over and over and over and over again. And so like in gourmets, it's not as bad, right? Because you want to eat them and you get a taste. So if you get one that grows really nice and beautiful, you don't mind recreating that 100 times and people eating it. But for me, like my personal philosophy is like if you're going for the medicinal compound, I think that environmental factor is going to play a huge role. So if you're using it to create a medicinal product, I would say grow multiple strains of it in different environments, so you have a little bit of diversity and you're not the same. Turkey tail, I just saw somebody doing it commercially. I've never really had luck. It takes a really long time to fruit. It takes a really long time to colonize. And again, it's out there a lot. You could find it a lot probably in your area. So the input, it might not be worth it. Like if you're somewhere in the world where turkey tail doesn't grow, I would say definitely give it your all. And like you could probably create some really amazing fruits and stuff. But again, it's like what's the end all goal, right? I mean, it's not a huge good food source, and when you're making medicines, you don't necessarily need it at a scale like that. I know there's like a lot of people, I guess, like, again, you know, it's just your end all goal, right? You don't want to, I don't eat turkey tail, but apparently you should eat turkey tail because it has tons of fiber in them. I've found out recently. I don't know how true it is. I keep trying to find this medical paper this guy's telling me about, but I am not having luck finding the evidence that turkey tail has tons of fiber. But if you've ever ground,
David, is turkey tail a polypole, so it grows on living trees?
it's a polyper. It's a saprophytic polyper, so it grows on dead and decaying trees, usually like oak, but I've seen it growing even, I think, on pine here, you know, like a Doug fir. So it's
Well,
not a picky mushroom at all, and all up and down the California coast, I imagine. Anywhere there's humidity, that mushroom will grow. Even here, this lady showed me a stump this year that I didn't think it grew. So I was fascinated. Again, you know, you want to grow the medicinals, make some medicine, and do cool stuff with it. But I would say if you don't have time to like go out, definitely work with like a local forager. You know, again, you're going to get the most diversity if you're like going for a medicinal product, which most people are. Like I heard people talking about mushrooms and cannabis. Like that's going to be a whole new thing. You know, see, this lady says it grows even on her willow tree. So Trametes is a very, very widely distributed mushroom. And so, I don't know. I wouldn't say I'm not trying to discourage you. It just takes a lot of input for not as good an output when you could like go run around or like say, hey, I'll give you this much money for your wild foraged ones. And if there becomes like, you know, bureaucracy involved that you have to have clean facilities and stuff, then find a really good cultivar local, maybe try to get it to somebody that does, like me, and I could put it on petri dish, and then that way you could just go get some like wood that's growing locally, and then we can just inoculate that wood, and then you can just set it up on your farm and grow it in a less passive way, right? So you're not putting in, you know, they grow really easily, right? So you can go find that local adapted species, see exactly what the wood, and now you have an abundance of this wood. Like for me, we don't have oak down here, right? But we got tons of aspen, we got tons of pine. So I need to go find these pine loving oysters and start growing them, so I'm not taking all these extra resources to create something that I could set up in a more natural system, right? In a syntropic way. So we're not taking as much, right? It's the idea of beyond organic. You're creating soil as you're taking from the soil.
Yeah, I think bringing it into a permaculture principle and perhaps taking some turkey tail spore and being able to cultivate it in your own backyard would be phenomenal. I tend to worry more that us as humans, once we find something it might be healthful or helpful, that we tend to over forage, and then we start losing some of the diversity within a natural. Yeah, we have a tendency of destroying habitats, and so not that I would want to see a lot of resources put towards it, but if we start finding out the benefits of some of these mushrooms in nature, we may over consume or over harvest.
Well, that's a very valid point, because there has been a lot of that in the Northeast Kingdom in Vermont, where they've now started to prevent people from harvesting chaga, especially chaga, because that one takes forever to get going, and the turkey tail, because both of them have legitimate cancer fighting properties. My beloved Pauline was on both of them. It's kind of cool that we have Dragonfly on next week, because they make a mushroom caplet that was part of Pauline's regimen, that has both the turkey tail, the chaga. I think it's got reishi. It's got a bunch of different mushrooms in it. So there's definitely some real value as far as medicinally. I don't know about the fiber. That I did not hear anything about. But yeah, there's a lot left to learn about these guys, for sure.
Yeah, and so I agree with you, over foraging, and that just becomes, again, I think educational platforms. Somebody was knocking on somebody, was like, oh, you're going to lead foraging classes, that's bad. I think no, I think ethics and like direction, because again, understanding the complexity of the system and saying that our intervention, yeah, maybe like going and tromping around the forest with 50 people and like machinery and stuff is definitely not a great way. But like if you can align with somebody who has a good ethics when they're foraging, it's a hard subject to say. But I think the more people that are informed, like it's like don't take more than you need for the year, and like if people can respect that and we can have that reciprocal relationship with nature, or even ask nature before you take, like don't just go taking things because you heard about it on a podcast and that somebody said you should go forage your own mushrooms. Yeah, I mean, there's a whole bucket to unravel, but it's like, a lot of people have this like controversy in foraging, especially with like gourmet mushrooms, that like, oh, you got to pluck versus pull, or you got to, and to me it's like, again, like we don't even quantify the understanding of this mycorrhizal system. The reason that it didn't come maybe 2 years after is not because a bunch of people picked it. Those mushrooms are going to come and go, and the amount that come and go that we don't see, like I've taken people out one day and the next day and walked by the same place 5 times and not seen the mushroom, and then all of a sudden I see the mushroom. It's just like to say that our intervention is like playing, it can go both ways, right? It's a double-edged sword. But like for me, in my own personal experience, I think the more people that are interacting with and engaging with it at a higher frequency, right, or like that forest bathing, like getting more people off of the AI screens and into like situations like this, there needs to be an educational platform based around it, and there needs to be information on it so people aren't doing things like that, and we're not worried about chasing a dollar when it comes to like a cure. And it's like, and so it's a really tough subject, but I see it all ways, and it sucks that we're like, oh, all of a sudden, turkey tail has these beneficial properties, and now we're gonna, but it's like, those turkey tails are doing something in the ecosystem, and they're making them, to me, they're fruiting because they want us to interact with them, and they want us to pick them, and they want us to take them. And like, is taking them not like also like spreading their spores maybe a little bit more? And like, I don't know, it does suck if you go in there with like machinery with no respect to Mother Nature, and you fuck, yeah, that's gonna fuck any ecosystem up, I believe. But like, even, I've led large groups, and I don't say everybody should know, but it's like, there's so much, like, it's like my secret spots. I'm like, do I really want to take these people here? And it's like, we all just left with a huge fucking basket, and the forest is just pumping them out every day, So it's like, it's hard to understand, right? It's like the pluck versus pull. People are like, you need to pull the mushroom, and then people are like, no, when you pull the mushroom, destroy the mycelial connection of the mushroom. And it's like, well, you got to chop it and leave it so it, like. And my thing, I look at people and I'm like, well, when you're harvesting an apple from a tree, do you pull the fucking apple off, or do you chop the apple off? Me personally, I pull the apple off because I want the whole piece of fruit, right? I don't chop it and leave a little bit. And it sucks because people that chop mushrooms, no offense to people who chop their mushrooms, but I'll go back the next day and I'll be like, yeah, mushroom. And it's like, oh, this is the part of the mycelium that you left here that you chopped, and it tried to grow, but its body was, half of its body's gone, but it's still unfolded its mycelial network, So like, I don't know, there's all kinds of like theories and double-edged swords to this thing. But yeah, I think that the more people, in my eyes, that are interacting with nature in a holistic way, I think one step closer to the solution, right? But
maybe that's a good segue into talking about the Southwest Fungi Fest and what you're doing there, and also with your lab that you're putting together.
Sure, yeah, totally. Thanks, Ken. Sure, yeah, creating that educational platform, it's it's been an interesting ride so far. I think 34 years of gathering information. I have a lot to share with people, and I want everybody in my region, the Southwest specifically, we don't get a lot of highlights of like what's actually happening here. And there's some amazing, brilliant people that I've got to interact with on this planet in this local region. And it's the Southwest Funga Fest because it's not just New Mexico, although I've been here my whole life and it's going to be here this year. It's in northern New Mexico, which is, a pretty sacred gem to the rest of the world. And I've got a little bit of pull from some people. I went as far as to say I was trying to gentrify the communities of northern New Mexico, and I was a white colonizer. It was the interesting social media, but really all I'm trying to do is create community, create an education platform that allows people in this region access to this information that they might not be able to reach without buying a $300 plane ticket, flying to the East Coast or the West Coast to a large conference and getting access to this information. Or, the closest one is Telluride, and I've been going for 10 years and it's getting pricey. We're doing some really cool stuff. We're not just encompassing mycology. Obviously, that's a huge push, but we're going to have our database collection site where any local farmer within the region, if they can't be here locally, they can mail it in, and we're going to send it to the database and start creating, a map of what's going on with the soil around this region. We're going to have Alan Rockefeller doing microscopy and photos. We're going to have local chefs doing mycology cook-offs. The whole theme of the festival is fungal preservation. So, I've really been in the fungal preservation window since I've been into mycology, and finding my passion project while making money and doing all these things is fun. So this is a good way to overarch it all. I'll have a couple other organizations pair with me. I've been the president of New Mexico Mycological Society, so I've not being an academic, I've been able to create a pretty good extensive network through academics by holding the title of president of the Mycological Society. And then people meet me and they're like, "You fucking guy," and I'm like, "Hey, here I am," And so we're trying to bring as many experts into this area. We got ladies like Rain Grant, who's the president of the Durango Psychedelic Society, who's going to be bringing, and she's also the owner of the Colorado Mushroom Company, and she's going to be bringing all her aspects of study. We got my friend down in El Paso, Mammalian Mycology, who's been providing access for veterans to this medicine and pairing with people all over California and Oregon to give veterans access to this sacred space that they need to create within themselves. We got he's also doing endophytic gene transfer research that he's going to be releasing to the public for the first time ever at the festival. That's being funded by a really well-known institute. I don't know right now because he hasn't told me all the details, but he's going to be unleashing all that endophytic gene transfer research that he's been doing, and he's gonna blow everybody's mind. And then we got William Padilla Brown, who's been doing RNA DNA stuff since the beginning. Dude taught himself how to sequence DNA in his basement during COVID when he was 26 years old. Matt Powers is gonna be there as well. We got Olga Sogas, hopefully from Smugtown Mushrooms, and then we got 20 local highlighters, collaborators. We got soil experts from this area. We got food experts. We got and best of all, we got the Spore Source, which is kids camp. I got 3 kids. I'm a dedicated father. We got a conscious kitchen where we have a really interesting group that goes around the festivals. They just did the Austin music festival, but you get 2 meals with your ticket price, all vegan, gluten-free, sourced by all local farms. We're going to be empowering all these local farms. I started teaching kids about mushrooms with this nonprofit called Access, and so I got a 5-week little course I did with the kids. I'm going to be teaming up with the local forest schools and hopefully privately contracting some of the workers to help me set up the Spore Source and do the 3-day kids camp. We're going to be having microscopes for kids to look through. I have a friend who's been working with game and fish in New Mexico for a really long time. I'm going to talk to him about opening up like a fishing portion for the kids, because we'll be partially on the Rio Grande. It gets kind of weird with getting licenses for all the kids, so I'm wondering if we're on private property in the Rio Grande, you know, and like the spore source is going to be amazing. I'm hoping to pair with this nonprofit called Kids Cook, which like goes into low-income families and helps provide food access and food safety for all the communities in America suffering, not just all over the world, right? Like I think it's 2 in 5 families have food insecurity in your own neighborhood. So don't forget that, people. Like, and so we're trying to bring all this with like drumming and breath. We have a local guy who's been on a liquid diet for the last 5 years, and he's narrowed his window of consumption down to an hour a day. He does breatharianism, qigong, so lots of energy work, and hopefully the land is going to be super sacred, and we're going to have some special stuff to offer there. They have like a natural amphitheater, an ancient archaic cave that's there on site, and we'll be doing tons of community restoration projects. We're going to try to get with the Rio Grande do water restoration projects. I mean, the list just goes on and on and on and on, and we're just barely getting started on like what we have to offer to the community, and like it'll be a pretty special place. So me and my buddy Julian are co-organizing it. We've both put in hundreds of hours of work for free, you know. So the guy who made the comment, I'll just stay it out there for everybody. We're not getting paid at this. We've been putting in endless hours of free work, hundreds of hours of free work. Nobody's colonizing or gentrifying any communities. I've been here for 34 years, working in and out of many communities, and I'm going to continue working with tons of other communities. We even have Marissa C. De Baca, who's president of New Mexico Psychedelic and Science Society, and like I said, the list just goes on. We'll have some music in the evening, but mostly community save the planet vibes over here. And then we just launched the gray area. It'll open in 2023. It's a community citizen science lab here at the Art Farm UnIncorporated. Oh, that's kind of my company. We got UnI Fungi, the gray area, and the Southwest Funga Fest are kind of all the projects I'm juggling at the moment. But the idea of the gray area is just, again, a community citizen science lab that anybody, open access. Obviously, it's not anytime you want. You just got to call up, make a reservation. I get approached by undergrads all the time who are, you know, studying, and they're geniuses, and they have all this information, but they don't want to spend the rest of their life in academia. And I come over here and I'm like, hey, well, it's kind of what we're doing. We're doing a little soil biology. We're just getting into all the DNA. We're about to buy our minion today, probably. I just keep slowly deciding, but it's like, what are you waiting for? Get in that lab, buddy. Start doing stuff. And so the idea is anybody can come. You can bring your family. Like for me, I didn't get to like consciously look through a microscope until I was at a mycological foray in my late 20s. I had a little kit microscope as a kid where you get prepared slides and look through them. I got one for my kids this Christmas just to try to get them to look through it. Other than that, it took to like 30 years before I got any of that exposure because I wasn't in academia and I didn't have access to it. Although I was like an alumni and I have a degree and I got my degree, it's just like access to a full-on lab to do like cool-ass citizen science research is not very few and far. I think there's like one in Oakland that Alan Rockefeller set up, and I think like Williams working on his shit out in Pennsylvania. But other than that, I don't know a lot of it going on. So like the idea is like, yeah, bring your whole family, come by on a Sunday. I got fucking 3,000 organic compounds you can come put on a slide and look in this microscope, and you guys can spend a couple hours learning some biological stuff. Or if you're a local mushroom farmer who's small scale and you need access to large pressure cookers and flow hoods, and like reserve your spot, come use the lab, come do what you need to do. Like that's the idea behind it, right? Of course I created this so I could go outside at any point in time and do all this cool shit and create the largest like library of genetics and plants and fungal in the entire Southwest while I'm doing it. But like, yeah, that's like what we're doing over here. So thanks for letting me plug that.
I think that was a brilliant plug too, man. Yeah.
Well, at that, it's 3 hours and 3 minutes, so it's probably time to wrap up. I know Av's got a busy schedule, and Ken, you just are an animal, so you probably need a break. And I got Paul over here, who's been very patient with me. So, David, thank you so much for sharing your time, your energy, your beautiful energy with us today, and keep us posted, my friend. Keep us posted.
Thanks,
Thank
David.
you all. Yeah,
Thanks, Leighton.
Thanks all. Thanks, Leighton. Thanks, Ken. And loving you all. We'll talk soon.
Yeah, thank you, Av, and Ken, too, guys. See you next week.
All right, guys, and don't forget we got Dragonfly Earth Medicine next week, so you're gonna want to be on with that, and that's a bunch more mushroom stuff too. So,
Absolutely.
all right, guys, with that, I'm gonna end the broadcast. Thanks everybody that was in the live chat today. Don't forget to pass this out to your friends, share it on your social media. The more people we get, the more we can progress and grow and save our planet. So let's do it, guys.
Yo.